Life's Instruction Manual. What genes are made of. So what is DNA? DNA is short for deoxyribonucleic acid, and it contains the plans for all the traits or characteristics in an organism. So if you remember, DNA is housed in the nucleus of a cell, and the nucleus contains the instructions for life, aka DNA. DNA, the genetic material. DNA inside all the genes for every body cell. A body cell has 2 times the amount of DNA as a gamete, egg, or sperm. So this goes back to mitosis and meiosis. So if you remember, somatic cells are body cells. Body cells have the total amount of DNA, or 2 times that of gametes or sex cells, which have half the amount of DNA, which makes sense if you think about it, because egg plus sperm, right, equals embryo or fertilized egg. What can DNA do? One, it can replicate before cell division. Remember mitosis, chromosomes need to replicate before they divide, because if the DNA doesn't replicate or copy itself, eventually you run out of DNA, right? If you keep splitting a cell in half without replicating what's inside, it's going to diminish by a half, then a half, then a half, get smaller and smaller and smaller. But if the DNA replicates first, the same amount of DNA is going to be inside of each successive cell. Number 2, DNA can also direct protein synthesis, so the making of proteins. It tells the ribosome to make a protein. So if you think back to a way long time ago, back to our cell unit, ribosomes were the site of protein synthesis. So DNA controls protein synthesis at the ribosome. So who discovered DNA? In 1869, Johann Friedrich Miescher is the first to isolate the DNA molecule. In 1952, Rosalind Franklin studies DNA molecule using X-ray diffraction. And in 1953, Watson and Crick developed the double helix model. So this brings us to the shape of DNA. DNA is a complex organic molecule. Yes, one of the organic molecules from biochemistry, right? It's a nucleic acid. And it has a double helix shape, which is like a twisted ladder. So if you think of a ladder, right, standing upright and then taking both ends and twisting them into a spiral, that's what a DNA molecule looks like. It consists of 2 phosphate sugar backbones, so alternating phosphate and sugar. The sugar is deoxyribose, and they are connected by a series of nucleotide base pairs. So here you can see the phosphate sugar backbone makes up like the rails of the ladder, and then the steps of the ladder, or the rungs, are the nucleotide base pairs. DNA nucleotide bases. So there are 4 bases in DNA: adenine, thymine, guanine, and cytosine. Okay, A, T, G, C. These bases are joined together in a specific way. So A always pairs with T, and G always pairs with C, and we call this base pairing. Okay, so this is kind of what the structure of DNA would look like. In between every base pair, okay, between every nucleotide base, you can see these red little lines. They're not like fused together, they're connected by bonds, and the bonds that hold them together are called hydrogen bonds. Hydrogen bonds are super weak, okay, which is, we'll find out in a minute, is really important to the process of DNA replication, or the copying of DNA. DNA replication is the process of making complementary copies of DNA through protein synthesis. So the first step in DNA replication, so in order for the DNA to copy itself, the first thing it has to do is unzip itself, we call it. Okay? And this is done with an enzyme called helicase, 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 and it unzips the DNA molecule. So what happens is the bonds between the base pairs are those weak hydrogen bonds we were talking about, and they can be easily broken with this enzyme. So the molecule will unwind and unzip itself, so it ends up with two sides of DNA. And it has all the information it needs to make a complementary pair. So you end up with two strands of DNA instead of the one. So if you look, we'll go back here, the helicase, the enzyme, would unzip in between here, okay? So you would have two sides now. Step 2 is protein synthesis. So DNA holds the code to make proteins. RNA transfers the code to the ribosome to copy the DNA. So we haven't talked about RNA yet. We will in a second. RNA is a smaller molecule similar to DNA, but it's single-stranded, and it actually brings the code to the ribosome. There are a few steps for protein synthesis: transcription, translation, and then complementary copies. So RNA is single-stranded, and another difference, so DNA is double-stranded, okay, it's a double helix. RNA is just single-stranded, it's one strand. RNA does not contain thymine like DNA, and instead it has a base called uracil, which bonds with adenine in RNA only. So DNA has A and T, adenine and thymine, and RNA has adenine and uracil, A and U. The major function of RNA is to help copy DNA by coding the chain and synthesizing proteins. And there's two types of RNA: messenger RNA, which we abbreviate mRNA, and transfer mRNA, transfer RNA, which we abbreviate tRNA. So transcription, the DNA message is copied into mRNA. So it goes from DNA, and you get the mRNA strand by complementary matching correct base pairs. So if you look, you have the one strand of DNA that we've unzipped, A, T, G, C, T, A, G, okay, whatever it may be. And then you get the complementary base pairs from the mRNA to create a strand. So A goes with U. Remember, there's no T in RNA. T goes with A, G goes with C, C goes with G, T goes with A, okay? And then you have translation, so the mRNA then tells the ribosomes which order to put together, and they do this in groups of 3. The tRNA matches the base pairs again and goes to get the amino acids from the cytoplasm. So all this is happening at once. Okay, these little tRNAs are going into the cytoplasm, and it's reading the code on the mRNA, and that code specifies a certain amino acid. And remember, when you line up amino acids, when you make an amino acid chain, that's how you make certain proteins. 2 complementary copies. Because of base pairing, each strand of DNA can replicate itself, making 2 new strands of DNA called complementary strands. All the work of DNA replication is done with the help of enzymes. The main enzyme is called DNA polymerase. From nucleus to ribosomes, this is what protein synthesis looks like. So DNA inside of the cell. Your mRNA is smaller, right? RNA is smaller, it's only single-stranded, so with transcription, it's a lot more efficient for a single-stranded molecule to move out of the cell and into the cytoplasm where translation can occur and the amino acid chain can be made, which then gets, made into a protein that codes for a trait. We have a universal code chart, and this is used to determine the specific amino acid sequence for a protein chain. So it's probably really hard to read on here. You can Google Universal Genetic Code Chart and it'll give you the same thing. So you use the bases, and that tells you which amino acid each group of 3 bases codes for. And these are just the abbreviations for the amino acids. Steps for using the chart: you need a DNA-based sequence of 3. You need to figure out the mRNA codon by matching the correct base pairs. This is mRNA, so you want to make sure you use a U, not a T. And then you find the 3 sequence code for the specific amino acid using the chart. So I'll show you how that works. So here we have an example DNA base sequence right here. You break into groups of 3, right? So every 3 bases, you break it off. So A, G, C, A. Okay, you can see that down there. And then what you have to do is match the mRNA base pairs. Okay, the chart uses RNA. So remember there's no T, so A is going to code with U, G codes with C codes with G, and so on, all the way through. And then you use the chart to find the matching amino acid for that mRNA codon you just created. So these are the abbreviations for the amino acids. Mutations. Sometimes mistakes can be made in the copying process of DNA. Any change in the original sequence of base pairs is called a mutation. And just an FYI, mutations can be good, mutations can be bad. Some mutations lead to the survival of a species, which then helps the species evolve over time. Others may kill a species. So what happens when a mistake is made in the replication of DNA? You can get albinism, right? This is a picture of a albino crocodile, alligator, ninja turtle, maybe. I forget what this is called, but when your fingers are like stuck together, they don't form correctly. Most mutations are not helpful, but every so often, a beneficial mutation occurs that allows a species to evolve. We have different types of mutations. Substitutions: one letter replaces another in a sequence. So example, A usually goes with T, but maybe G accidentally pairs with A. Okay, that's called the substitution. One letter is substituted for another. Insertion is when one letter is added to the sequence, so an extra letter that doesn't belong. And a deletion is when a letter is removed, so it's the opposite when a letter is removed, so there's one less letter than needed. So here are just some examples. Here's a substitution. Okay, an insertion, right? It's one more base pair long, and a deletion, it's one base pair short.